Hello friends, today we are going to discuss on the topic overview of bacterial cell. Today we will try to learn about size, shape and arrangement of bacterial cells. To get an insight about a typical bacterial cell, cell membrane, cytoplasm matrix, inclusion bodies and nucleoid. To know about the structure of cell wall and difference in gram-positive and gram-negative cell wall. To understand the need of capsule, flagella and its mechanism of movement. Lastly, to know about bacterial endospore. Let me first introduce about world of bacteria. Bacterium, a single cell prokaryotic organism, is extremely small in size and simple in structure, which lack chlorophyll pigments. A bacterial cell has cell wall but lacks membrane bound nucleus. There are thousands of different kinds of bacteria which live in every conceivable environment all over the world. Stewart Brand rightly said about bacteria, if you don't like bacteria, you are on the wrong planet. Bacteria adapt to become well suited to their environments and therefore come in many shapes and forms. At the outset, let us learn about size, shape and arrangement of bacterial cells. The size, shape and arrangement of bacteria and other microbes is the result of their genes and thus is a defining characteristic called morphology. Bacteria come in a puzzling and exciting variety of size and shapes. The unit of measurement used in bacteriology is the micron, and 1 micron or micrometer equals to 1,000th of a millimeter. Depending on their shape, bacteria are classified into several varieties. Cocci, from cocos meaning berry or spherical or oval cells. Bacilli, from bacillus meaning rod or rod shaped cells. Vibros are comma shaped curved rods and derived their name from their characteristic vibrative motility. Spirilla are rigid spiral forms. Spirochetes, from spira meaning coil and chaetae meaning hair, are flexose spiral forms. Actinomycetes are branching filamentous bacteria, so called because of a fan-side resemblance to the radiating rays of the sun when seen in a tissue lesions, from actis meaning ray and mycus meaning fungus. Mycoplasma are bacteria that are cell wall deficient and hence don't possess a stable morphology. They occur as round or oval bodies and as interlacing filaments. Bacteria sometimes show characteristic cellular arrangement or grouping. According to the plane of cellular division, diplococcus occurs when a coccus divides along a one plane and arranges in pairs as found in Acinetobacter brucella. Streptococci occurs when a coccus divides along one plane and arranges like a chain in Streptococcus agalacti, Streptococcus aeruginosa, Streptococcus bovis. The individual coccus undergoes division along 3 perpendicular partitions so that multiplication takes place in 3 directions, giving groups of 8 cells called as sarcina. If a coccus divides along multiple planes and arranges as grape-like clusters, it is called Staphylococcus, like Staphylococcus aureus. Tetrad is a group of 4 cells formed from a coccus which divides along a perpendicular planes and arranges like a square. Bacterial structure. The outer layer or cell envelope consists of 2 components, a rigid cell wall and beneath it is a cytoplasmic or plasma membrane. The cell envelope encloses the protoplasm comprising the cytoplasm inclusions such as ribosomes and mesosomes, granules, vacuoles and the nuclear body. Cell wall. Beneath the external structure is the cell wall. It is very rigid and gives shape to the cell. Its main function is to prevent the cell from expanding and eventually bursting due to water uptake. Cell wall constitutes a significant portion of the dry weight of the cell, and it is essential for bacterial growth and division. Chemically, the cell wall is composed of peptidoglycan, mucopeptide, peptidoglycan or murin, formed by normal acyl glucosamine and normal acyl muramic acid, alternating in chains, cross-linked by peptide chains. Embedded in it are polyalcohol called teichoic acids. Some are linked to lipids and are called lipoteichoic acids. Lipoteichoic acid link peptidoglycan to cytoplasmic membrane, and the peptidoglycan gives rigidity. Outer membrane. Outer membrane is found only in gram-negative bacteria. Its function as initial barrier to the environment, and it composed of lipopolysaccharide and phospholipids. Lipopolysaccharides. The lipopolysaccharides present on the cell walls of gram-negative bacteria account for their endotoxic activity and antigenous specificity. A bacterium is referred as a protoplast when it is without cell wall. Cell membrane. A cell is the basic unit of life, and all organisms are made up of one or many cells. One of the things that all cells have in common is the cell membrane. It is a barrier that separates the cell from the surrounding environment. This outer boundary of the cell is also called the plasma membrane. It is composed of 4 different types of molecules. First, phospholipids, cholesterol, proteins, carbohydrates. Phospholipids. The fundamental building blocks of all cell membranes are phospholipids, which are amphipathic molecules. A single phospholipid molecule has 2 different ends, a head and a tail. The head end contains a phosphate group and is hydrophilic. This means that it likes or is attracted to water molecules. The tail end is made up of 2 strings of hydrogen and carbon atoms called fatty acid chains. These chains are hydrophobic or don't like to mingle with water molecules. The phospholipids of a cell membrane are arranged in a double layer called the lipid bilayer. The hydrophilic phosphate heads are always arranged so that they are near water. Watery fluids are found both inside a cell, intracellular fluid, and outside a cell, extracellular fluid. Lipids constitute approximately 50% of the mass of most cell membranes, although this proportion varies depending on the type of membrane. Lipid composition of cell membranes. An important property of a lipid bilayer is that they behave as 2-dimensional fluids in which individual molecules, both lipids and proteins, are free to rotate and move in a lateral directions. Mobility of phospholipids in a membrane. Individual phospholipids can rotate and move laterally within a bilayer because of its hydrocarbon ring structure. Cholesterol plays a distinctive role in determining membrane fluidity. Cholesterol molecules insert into the bilayer with their polar hydroxyl groups close to the hydrophilic head groups of the phospholipids. They strengthen the membrane by preventing some small molecules from crossing it. Now the carbohydrates. Carbohydrates or sugars are sometimes found attached to proteins or lipids on the outside of the cell membrane. That is, they are only found on the extracellular side of cell membrane. Together these carbohydrates form the glycocalyx. Now the membrane proteins. Proteins are other major constituent of cell membranes, constituting 25 to 75% of the mass of various membranes of the cell. The current model of membrane structure proposed by Jonathan Singer and Garth Nicholson in 1972 views membranes as a fluid mosaic in which proteins are inserted in a lipid bilayer. While phospholipids provide the basic structural organization of membranes, membrane proteins carry out the specific functions of the different membranes of cell. These proteins are divided into two general classes. First, integral proteins located in the lipid bilayer and peripheral proteins outside of the lipid bilayer based on the nature of their association with the membrane. Fluid mosaic model of the membrane structure. Biological membranes consist of proteins inserted into a lipid bilayer. Integral membrane proteins are embedded in the membrane usually via alpha helical regions of 20 to 25 hydrophobic amino acids. Many integral membrane proteins called as transmembrane proteins span the lipid bilayer with portions exposed on both sides of the membrane. The hydrophobic side chains of these amino acids interact with the fatty acid chains of the membrane lipids, and the formation of an alpha helix neutralizes the polar character of the peptide bonds. Proteins can also be anchored in membranes by lipids that are covalently attached to polypeptide chain. Distinct lipid modifications anchor proteins to the cytosolic and extracellular phases of the plasma membrane. Proteins can be anchored to the cytosolic phase of the membrane either by the addition of a 14-carbon fatty acid, that is mysteric acid, to their amino terminus, or by the addition of either a 16-carbon fatty acid, that is palmitic acid, or a 15 or 20 carbon prenyl groups to the side chains of cysteine residues. Permeability of the phospholipid bilayers. Small uncharged molecules can diffuse freely through a phospholipid bilayer. However, the bilayer is impermeable to large polar molecules such as glucose and amino acids and to ions. Although ions and most polar molecules cannot diffuse across the lipid bilayer, many such molecules such as glucose are able to cross cell membranes. These molecules pass across membranes via the action of specific transmembrane proteins, which act as transporters. Such transport proteins determine the selective permeability of cell membranes and thus play a crucial role in membrane function. They contain multiple membrane spanning regions that form a passage through a lipid bilayer, allowing polar or charged molecules to cross the membrane through a protein pore without interacting with the hydrophobic fatty acid chains of the membrane phospholipids. Channel proteins from open pores through the membrane, allowing the free passage of any molecule of the appropriate size. Ion channels, for example, allow the passage of inorganic ions such as sodium, potassium, calcium and chloride ions across the plasma membrane. Once open, channel proteins form small pores through which ions of the appropriate size and charge can cross the membrane by free diffusion. The pores formed by these channel proteins are not permanently open. Rather, they can be selectively opened and closed in response to extracellular signals, allowing the cell to control the movement of ions across the membrane. In contrast to channel proteins, carrier proteins selectively bind and transport specific small molecules such as glucose. Rather than forming open channels, carrier proteins act like enzymes to facilitate the passage of specific molecules across membranes. The plasma membrane of the cell has two main roles. It is a physical barrier and it regulates the exchange of materials with its surroundings. Cytoplasmic matrix. Cytoplasm is everything inside the cell, excluding nucleus. Cytoplasmic matrix or cytosol or intracellular fluid is the liquid found inside cells. It is separated into compartments by membranes. In prokaryotes, most of the chemical reactions of metabolism takes place in the cytosol, while a few takes place in membranes or in the periplasmic space. The cytosol is a complex mixture of substances dissolved in water. It accounts for about 70% of the total cell volume. It is a gelatinous substance consisting mainly of cytoskeleton filaments, organic molecules, salt and water. Chemically, the cytoplasmic matrix is composed of many chemical elements in the form of atoms, ions and molecules. Of the 92 naturally occurring elements, approximately 46 are found in the cytosol. 24 of these are essential elements, while others are present in cytosol only because they exist in the environment with which the organism interacts. Of the 24 essential elements, 6 play especially important roles in living systems. The concentrations of ions such as sodium and potassium are different in the cytosol than in the extracellular fluid. These differences in ion levels are important in processes such as osmoregulation, cell signaling and the generation of action potentials in excitable cells such as endocrine, nerve and muscle cells. Now the inclusion bodies. Inclusion bodies are nuclear or cytoplasmic aggregates of sustainable substances, usually proteins. They typically represent sites of viral multiplication in a bacterium or a eukaryotic cell and usually consist of viral capsid proteins. Mechanism of formation. When genes from one organism are expressed in another, the resulting protein sometimes forms inclusion bodies. This is often true when large evolutionary distances are crossed. For example, a cDNA isolated from eukarya is expressed as a recombinant gene in a prokaryote, risks the formation of the inactive aggregates of protein known as inclusion bodies. Now, the nucleoid. The nucleus is not distinct and has no nuclear membrane or nucleolus, and the genetic material consists of DNA. The nucleoid, meaning nucleus-like, is an irregular shaped region within the cell of a prokaryote that contains all or most of the genetic material called gonophore. In contrast to the nucleus of the eukaryotic cell, it is not surrounded by a nuclear membrane. Now, the cell wall peptidoglycan structure. The cell wall surrounds the plasma membrane and protects the cell from changes in water pressure. The bacterial cell wall consists of peptidoglycan, a polymer consisting of normal style glucosamine NAG and normal style muramic acid NAM and short chains of amino acids. Structure of peptidoglycan in a cell wall. Alternating NAM and NAG molecules form a carbohydrate backbone. Rows of NAG and NAM are linked by polypeptides. The structure of the polypeptide cross bridges may vary, but they always have a tetrapeptide side chain, which consists of 4 amino acids attached to NAMs. The amino acids occur in alternating D and L forms. While peptidoglycan is present in most of all bacterial cell walls, there are 2 basic variations of structure seen in most bacterial cells. One described as gram-positive and one described as gram-negative. Gram-positive cell wall. Gram-positive cell walls consist of many layers of peptidoglycan and also contain teichoic acids. Teichoic acids may bind and regulate movement of cations into and out of the cell, prevent extensive wall breakdown and possible cell lysis. Now the gram-negative cell wall. Gram-negative bacteria have a lipopolysaccharide, lipoprotein, phospholipid, outer membrane surrounding a thin, sometimes a single peptidoglycan layer. Gram-negative cell walls have no teichoic acids. The outer membrane protects the cell from phagocytosis and from the penicillin, lysozyme and other chemicals. Porins are proteins that permit small molecules to pass through the outer membrane. Specific channel proteins allow the outer molecules to move through the outer membrane. The lipopolysaccharide component of the outer membrane consists of sugars that function as antigens and lipidae, which is an endotoxin. Endotoxin causes fever and shock. Now we will discuss capsule. Capsule is the outermost layer of the bacteria. It is a condensed, well-defined layer closely surrounding the cell. They are usually polysaccharide, and if polysaccharide envelopes the whole bacterium, it is capsule, and their production depends on the growth conditions. They are secreted by the cell into the external environment and are highly impermeable. When it forms a loose mesh work of fibrils extending outward, it is glycocalyx, and when masses of polymer that form appear to be totally detached from the cell, and if the cell are then entrapped in it, are described as slime layer. The capsule protects against complement and is anti-phagocytic. Now the flagella and motility. A flagellum is a lash-like appendage that protrudes from the cell body of certain prokaryotic and eukaryotic cells. The word flagellum in Latin means whip. The primary role of the flagellum is locomotion, but it also often has function as a sensory organelle, being sensitive to chemicals and temperatures outside the cell. Flagella are long hair-like helical filaments extending from cytoplasmic membrane to exterior of the cell. Flagellin is highly antigenic and functions in cell motility. The location of the flagella depends on bacterial species, as polar situated at one or both ends, which swims in back and forth fashion, and lateral along the sides. Now the pili or fimbriae. Hair-like proteinaceous structures that extend from the cell membrane to external environment are pili, which are otherwise known as fimbriae. They are thinner, shorter and more numerous than flagella, and they do not function in motility. The fimbriae is composed of a subunit called a pili. There are two types of pili, namely non-sex pili, that is common pili, and the sex pili. The fimbriae are antigenic and mediate their adhesion, which inhibits phagocytosis. The sex pili help in conjugation. Mechanism of flagellar movement. The bacterial flagellum is driven by a rotatory engine, the Mott complex, made up of protein located at the flagellum's anchor point on the inner cell membrane. The engine is powered by proton motive force, by the flow of protons across the bacterial cell membrane due to the concentration gradient set up by the cell's metabolism. In Vibrio species, there are two kinds of flagella, lateral and polar, and some are driven by a sodium ion pump rather than a proton pump. The rotor alone can operate at 6,000 to 17,000 rpm, but with the flagellar filament attached usually only reaches 200 to 1000 rpm. The flagellum is highly energy efficient and uses very little energy. The cylindrical shape of flagella is suited for the locomotion of microscopic organisms. The organisms operate at a low Reynolds number, where the viscosity of the surrounding water is much more important than its mass or inertia. Now we will discuss the bacterial endospore. Some bacteria have the ability to form highly resistant resting stage called spores. which helps them to overcome adverse environmental conditions that are unfavorable for vegetative growth of cell produced by certain bacteria from the Firmicute phylum. The name endospore is suggestive of a spore or seed-like form. Endo means within, but it is not a true spore. In endospore formation, the bacterium divides within its cell wall. One side then engulfs the other. Endospore enables bacteria to lie dormant for extended periods, even centuries. When the environment becomes more favorable, the endospore can reactivate itself to the vegetative state. Structure of endospore. Bacteria produce a single endospore internally. The spore is sometimes surrounded by a thin covering known as the exosporium, which overlies the spore coat. The cortex lies beneath the spore coat and consists of peptidoglycan. The core wall lies beneath the cortex and surrounds the protoplast or core of the endospore. The core contains the spore chromosomal DNA, which is encased in the chromatin-like proteins known as SASPs, small acid-soluble spore protein that protect the spore DNA from UV radiation and heat. The core also contains normal cell structures such as ribosomes and other enzymes, but is not metabolically active. Location of endospore. The position of the endospore differs among the bacterial species and is useful in identification. The main types within the cell are terminal, subterminal, and centrally placed endospores. Thank you so much for being with me during this lecture.